So, welcome, Kat. Can you give yourself a little bit of an introduction?
Sure, yeah. I'm Kat Hartwig, the Executive Director of Living Lakes Canada. And I'm
Lovely.
keen to see how we set up study designs.
Well, it's so great to see you here. I'm really excited about the work we're doing together. So, I'm going to show you and answer to your request what I typically do, what goes behind the scenes, if you like, on good design on an eDNA project. So, this is a survey protocol for Pacific giant salamander. So, for this purpose, we're actually designing a survey that we will use to go and survey for the species in the Chilliwack, well, within its home range. So when I'm asked to start, I have to basically write a project background to say why we're doing it, and project objectives, and then a species account. And the species account details the ecology of the species, and it's really important. So I explain to people that eDNA is a marriage of disciplines. It's a discipline of ecology. You have to wear your ecologist hat and understand the species that you're working on. You have to be an expert. It's also, you have to be a bit of a hydrologist to understand how water might move through a system and where you might best detect the DNA from your focal taxa. And you also have to be a bit of a geneticist, and you have to do all the genetic work appropriately with the lab. So you need to wear all three hats. So that's what we're doing here. So this is the ecology section, talking about the ecology of giant salamander. And let's talk about how, or some good sources of reference. So on my right screen here, to become a mini species expert, or at least qualified, what we need to do is learn as much as we can about giant salamanders so that I can start populating survey points here on this map on the left. And so there's several tools that are available to us as ecologists or biologists to do this. One of the best sources of one-stop shopping is actually the recovery strategy. So that's a series produced by Environment and Climate Change Canada in cooperation with the provinces, and usually in cooperation with a panel of experts that are engaged on writing the document. So the recovery strategy documents, have you used them before in your work? Are you familiar with them?
We are familiar, yes.
Perfect, yeah. So the recovery strategy documents just take you through all the basic the basic biology of the species, and I understand we're not going to try to cover all the content, but there's information here on movement and dispersal that's very important for me, so I understand how they're moving, and there's information on habitat, and I won't bother finding it in this document, but that information is really key. That allows me to make sure I'm sampling in the right locations. So that's a great place to start, is those recovery strategies. Sometimes there's extra accounts or other information, so I sort of scour the earth looking for things. So the Identified Wildlife Management Strategy series is great. It gives me a lot of management information, tells me about the habitat requirements for the species, the life history, so I can learn things like when is it in the stream, when is it reproducing, all of those ecological parameters that are very important for design. We're also actually designing an experiment that's assessing drought conditions. So I found a paper, I go to the primary literature, and I found a paper that looked at use in hyporheic zones. And so those are the areas that are substrate flood, like still wet. When the stream dries out, the salamanders might be able to survive there. Mm-hmm. And so this is a paper written in California that looked at survival in the hyporheic zone, so in desiccated streams. I'm also concerned about the effects of logging. So I go to the primary literature and I find papers, and this is just an example of such, but I find papers on logging and how that affects movements and survival and persistence of giant salamanders. And not surprisingly, it's not a good news story. Logging is very bad for them. So yeah. And then finally, I went out in the field last year, and I inventoried streams to assess which streams were dry and which streams were wet. I assessed 183 streams, and I found 68% of them were dry, which was a very alarming year. We had, what did you call it yesterday? A micro drought or something?
Flash droughts.
A flash drought.
Yeah.
So we had a flash drought event in Chilliwack where it only received 2/3 of its average annual precipitation in the summer season. Mm-hmm. And so that's worrisome for my client. And so they want me to do some research to figure out how that flash drought might have affected giant salamanders. And so I went out there and I collected samples of giant salamanders, swabs, to help develop the primer, the genetic tool, to work on the species. So that's some of the inventory I'll pull together. And I'll just leave that up on this and bring up my protocol on this and talk a little bit about site selection. So this whole section of the report is dedicated to site selection, and it talks about some of the layers I use. So rather than reading it to you or boring you with the details, I'm just going to start bringing those layers on in a GIS interface. So you can do GIS in something as simple as Google Earth or Google Maps and just start looking at ortho images, but you can get a lot more powerful if you start using apps like ArcMap or QGIS, which is a free app, to access some of the spatial layers. Biology is, after all, the study of patterns. Whether you're looking for something as simple as an owl camouflaged against the trunk of the tree and you see the pattern break in its feathers against the bark, and that's discerning a pattern. That's a very simple level of pattern study. But at a landscape level, biologists are constantly looking for patterns in the distribution of species, and it's all spatial. And so if biology is a study of patterns and you're looking at landscape scale patterns, it really benefits you as a biologist to understand GIS. So that's why I use GIS as a very powerful tool. I would say it's almost a necessity for doing some of this work. So let's start turning on some of those layers in GIS. So the first thing I want to understand is land ownership, to make sure I stay within my client's constraints with funding and I don't trespass onto private land if that's out of scope. So the first thing I'll do is turn on parks. If I go into parks, I have to have a park use permit for doing research in a park, and that includes federal and provincial, but there are no federal parks in here, so I just turn on provincial. If I intend to work on First Nations lands, I really need, and this is not the unceded, these are First Nation reserves, but the unceded territories are understandably bigger. But I need to understand where First Nation reserves are. So the other layer I like to use is private land. And so I like to know if I'm trespassing on anyone's property, then I need to understand where I might have to ask for landowner permission to access different streams. So there's my land tenure information. I also need to think about access. So I've turned on forest roads. They're so faint on my screen it doesn't show at the scale, but they are. If I were to zoom in, you can see the Forest Service roads coming in there. So that tells me a little bit about access. That helps me with budgeting, with planning, knowing how many days it's going to take to get to this many sites. And so I have to always stay within a budget that's usually imposed upon me by the client or the project limitations. But understanding access and estimating the time required to do these gets me through that.
So how many sites would you do in a day, do you think?
Oh gosh, that's a good question. And it's really important from a budgeting perspective to understand that. So I average, my golden rule is about 8 sites a day. In the early days, I used to push it and I did as many as 18 sites one day.
Wow.
But I was collecting from 7 a.m. We went out early from the hotel, went out and collected all day, got back at about 8 p.m. at night. Long day, we were exhausted. But then I've got 18 sites to filter that night because I have to filter them within 24 hours of collection. So we were filtering till 2 or 3 a.m. And it was exhausting. And that was my field schedule for the whole week because that's what I'd accidentally budgeted for, not accounting for the time it takes to filter. So now what I do is my guidance, and I ask or suggest it to other people, budget for about 8 sites a day. That's about 8 hours of collection. And I know what you're thinking, 8 hours, that's pretty wimpy for a field day. You should do better than that. But I'm only halfway through. I've still got to do my filtration. And so it turns into a 12 or 14 hour field day if you're doing 8 sites. So by the time you get your filtration done. So that's a great question. So about 8 sites a day is what we can do. So I know my budget. I know how much it costs per day to be out there with the technician, vehicles, everything else. And I can work backwards to how many sites I can possibly fit in. But that 8 sites is a golden rule. So yeah. So let's keep going on the information. But that was a really insightful question. So you saw that I mentioned earlier that a giant salamander depends on old growth. It's an old growth dependent species. It's affected badly by logging. And so I also pulled the forest cover data and I queried out by age class, and I selected age class 8 and 9, so 100 to 140 years and 140 years and greater. So we learned from this paper over here that clear-cut logging is bad for giant salamanders, and we know this is true for a lot of species, spotted owls, marbled murrelets, etc. And so in answering the question or designing a study for giant salamanders, it behooves you to actually understand what is available for old growth. So I queried what's called the vegetation resource inventory layer. I'm looking for my mouse. The vegetation resource inventory layer, which is forest cover data, and I've colored up what is old growth in green here, 8 and 9.
Wow, there's not very much old growth.
No, and we might wonder where it went. So let's look at logging depletion. So these are my depletion layers. So the light brown with the purple outlines is showing you older logging that was in previous years. The red are cut blocks that were just logged last year alone.
Wow.
So that's how much habitat we're losing every year within the range of just one species. So it really is a detrimental activity. Yeah. So when you ask your question, where's the old growth gone? You can see the overlap now. Wherever it's not green, it's been logged.
Wow.
And so that makes it very restrictive for me to map my giant salamander sites because I need to sample in old growth. They're gone from clear cuts. So to that end, I brought in a bunch of data from a bunch of different layers. The first thing I brought in was the critical habitat. That's publicly available. All those orange streams are mapped by the federal government as critical habitat for giant salamander. The next thing I brought in is wildlife habitat areas. These were mapped by the province for conservation of giant salamander. This is estimated to be less than 26% of the range of the species. So the province has a long way to go to finish protecting the species properly. Yeah. And on critical habitat on federal land, it's afforded automatic protection, but it's up to the province to implement effective legal protection where critical habitat occurs on Crown land. So the province, again, has a long way to go with respect to protecting all the critical habitat as well. So we've got some tools, but the province is under implementing or underperforming. So
it looks like agencies aren't talking to each other.
It does, and it's a big problem. As you know, multi-jurisdiction cooperation really fetters conservation. So let's bring in the final layers here, which is the giant salamander data. So every red dot I just turned on, and I'm going to turn off those distracting clear cuts. So now we just have old clear cuts. I don't have last year's because it got too distracting. I have the red dots are giant salamander observations, the orange is critical habitat, and the green is WHAs. Now, what I'm going to do is start to think, where can I find giant salamanders? There's one other little piece of information that's important. In reading through that recovery strategy document, we learned that giant salamanders and coastal tailed frogs actually have a lot of overlap. So I also brought in all of my data for coastal tailed frog. So this gives me some ideas about where I might look for giant salamander outside the known range, because I know from the recovery strategy that it's under inventoried. People have never even looked here. And so I need to start thinking about that for my client or in my inventory. I'll finally turn on, I'm not going to actually bother turning on my stuff from last year. There we go. So the final thing I'll do is turn on, and this is getting really cluttered, the streams that were wet or dry last year. Red is dry, bone dry, and blue is wet. When all of that's considered, I can use that information to go through and map out where I want to sample ED. Considering all of those layers, so we looked at clear cuts, we looked at available old growth forest, because I know that's where they're going to be. I considered critical habitat, and I considered provincial protections under Forest and Range Practice Act like WHAs. So considering all of that information, I knew where I could survey within the range. To go outside the range, I considered tailed frog detections to tell me, because there are some Patrick species where suitable habitats might exist outside the range that have never been surveyed. And it's really exciting. I'm going to use a really powerful tool, eDNA, that we've been talking about, to actually go and achieve survey in these areas. I'm also going to use it on the long term to start testing some of those dry streams and look at recolonization rates to see if they could survive in those hyperrheic zones, in those dry substrates. And so that's our multi-year project. In year one, we're just going to be range expanding and testing the efficacy and using eDNA. So let's just zoom in on how I did, and we're pretty much finished our explanation, but all of this information stacks up, and if I zoom in, this is an example of a kind of creek I want to look for. I've got a bit of age class 7 and some age class, 8 and 9, so I've got some old growth forest, I've got a stream running through it, it has not been identified as critical habitat, or it would be orange, and it has not been identified as a WHA, or it would be light green. So this is a perfect example of a stream that is very likely to contain giant salamanders, yet it is unprotected at a federal level or at a provincial level. And so if I can go in there with eDNA and find giant salamanders, I will add conservation benefit by allowing or being able to rationalize the need for protection at the site. So that's basically the whole story. As you can see, site selection is not a simple business. If I had started all of this exercise and just asked you to go through and map sites with none of these layers and not given you any of this information that I compiled over the course of several days, I'll leave my streams on, none of the logging, none of the VRI, and none of the parks or First Nations or land tenure. And I just said to you, great, Kat, here you go, blank slate, giant salamanders found in here. I want you to efficiently design a survey. You would be in a lot of trouble. You wouldn't know where to start. Exactly. So we have to start layering up the information so that we have a really good, powerful, efficient, and effective survey design that utilizes this method properly. So thanks for listening. Did that all make sense?
Yeah, it did. Thanks for the explanation. It's a lot of work.
It's a lot of work. People underestimate, but if you don't design, if you don't prepare, you're going to fail. Exactly. And so you really have to prepare properly when you do an eDNA experiment. Not only does you have to wear the 3 hats, ecologist, hydrologist, and geneticist, but you also have to be really good at the ecology job of pulling everything together. So a lot of people call me for help with this aspect, but as you can see, there's a lot to it.
There is, yeah.